Welcome to another tutorial of video series on genome wide analysis. In this video, I will be showing now how we can select the protein families based on our research area and interest. In the previous video, we have discussed the interpretation and to-do list of genome wide analysis. Now, we will be formally starting genome wide analysis with the selection of a protein family. So let's begin. So here you can see whenever we want to select our protein family for genome-wide analysis, so what we have to do, first we have to focus on what is our research area or what is our niche of research. So how we can identify, so we can identify with the help of like in which lab we are working and whether we are working on plants or whether we are working on animals. So here you can see it depends on what is your research group and what is your own interests. So if you belongs to plant sciences, so you will definitely studying the different mechanism of plants, and if you will working on human biotechnology and like you are working on cancer, so it depends on your own interest. So first of all, you can study through literature. I mean, wherever you are working and what is their research interest, you can study about the research topic. Let's suppose if you are working on a plant stress, so you can find out some literature that what are those families of proteins which are involved in plant stresses, so you can find them through the literature. So another way is through the chat gpt. So let's suppose if you are working on the nickel or the cadmium stress, so you can simply ask to the chat gpt that what are those families of proteins which are involved in regulating the stress involvement of cadmium or other heavy metals. So similarly you can use chat gpt, and you can also ask your supervisor that if he wants to give you any specific topic, so of course you can also start with that one. After that, you can also ask your senior. You can search them through online different databases, like you can find them on the researchgate or the different social media like Twitter. You can use them to find your relevant research seniors so that they can help you to guide what topic you should select for genome wide analysis. So let's suppose if your research area belong to plant sciences and specifically you are working on the salt stress. So here I have listed the 10 gene families and the protein families which are involved in the salt stress. So you can select any of them depending upon your own need. So here you can see I have listed SOS, that are the salt overly sensitive proteins pathway. Similarly, LEA, late embryogenesis abundant protein. Similarly, high affinity potassium transporter. And similarly, you can also work on the thylene responsive factor, superoxide dismutases. Yeah, these are the antioxidant enzyme. You can work on this. And similarly, for heat shock proteins, mitogen activated protein kinases, aquaporins. Similarly, proline biosynthesis enzyme and respiratory burst oxidase homolog proteins. So you can select any of them if your area belongs to the salt stress. But here I have listed only 10, and I will also give you this file in the form of PDF, and I will upload this file to my drive and I will paste its link in this video description, so you can also use this. But here you can see I have listed only 10, so you can also select more by studying through the literature. So similarly, if your area is cold stress, so here you can see I have listed 10 gene protein families which are involved in the cold stress. So among them, the CBF are the DREB proteins, which are dehydration responsive elements, antifreeze proteins, similarly cold shock proteins, and cold regulated proteins, ice binding protein, glycerol 3-phosphate dehydrogenase, and lipid desaturases, chaperons, ice-bonds, and antioxidant enzymes. They are involved in cold stress. So what you need to do, you need to only select one, and we will take it to the next step to download their sequences from the different databases. So similarly, if you are working on the drought stress, And similarly, if you are working on the high temperature stress, so although they are different, but most of the proteins are common, like the DREB, heat shock proteins, LEA proteins, aquaporin, ROS scavenging enzyme, abscisic acid, RBOH, dehydrins, MAPK, and apoptosis related proteins. So here you can see if you are working on the light and shade stress, so you can choose phytochrome, cryptochrome, photoreceptor, light harvesting complexes, and chlorophyll binding proteins. Similarly, phytochrome interacting proteins, COP1, similarly hypocotyl elongated 5, and repair protein, xanthophylls, and carotenoid binding proteins. So all of these proteins are involved in light and shade stress. So you can select any of these from this list as well. So if you are working on the pathogens, so you can select like TLR, NLR, similarly R proteins. You can work on pattern recognition receptors. Similarly, you can work on interferons, antimicrobial peptides, and major histocompatibility complexes, cytokinins, complement protein, phagocytic receptors, antibodies, and proteases and peptidases. So if your work is related to the nutrient deficiency, so you can work on like iron regulated protein, transporter protein. You can work on myoinositol 1-phosphate synthase, phosphate transporters, nutrient sensor kinases, similarly phytochelatins, and nutrient responsive transcription factor, sulfate transporter, ammonium transporter, similarly metallothioneins, similarly ferroportin, and calcium sensor and channels. So if your research area is related to finding anti-cancer protein, so you can definitely work on tumor suppressor protein, oncogene, similarly receptor tyrosine, and so on. Here you can see we have also checkpoint proteins, and we have NU receptor kinases, heat shock proteins, and poly-ADP ribose polymerase, and similarly P21 activated kinase telomerase, angiogenesis inhibitors, and tumor antigens. So once you have selected your protein, so it is important to make sure through the Google Scholar if this protein family is already published. So whatever you are going to select, make sure that this protein should be novel. It means that if this protein is already published in any article, so there is no use to again studying the genome wide association analysis. So make sure to check whatever you are selecting. Please make sure whether this protein or its genome wide characterization is already being done or not. So your selection should be a novel so that it can be published in good journal. So hopefully this list will be helpful in selecting the gene protein family for your genome wide analysis. So if you have still any question in selection, please let me know in the comment section. Thank you for watching. Bye bye.